The receptor tyrosine kinase, EphA2, binds to its ephrin A1 ligand, which is found at the membrane of opposing cells. EphA2 thus binds to ephrin A1 at cell-to-cell contacts, and it causes cell repulsion through activation of intracellular signaling pathways that control cytoskeletal dynamics. As a result, this signaling axis controls contact-dependent cell communication that drives cell adhesion, migration, morphology, and survival. Misregulation of the EphA2 ephrin A1 signaling axis causes aberrant cell-to-cell contacts that contribute to metastasis. Solid tumors are characterized by an acidic extracellular medium. We took advantage of this tumor property to create type 7, a transmembrane ligand for EphA2. Type 7 is a peptide obtained by adding glutamic acid residues to the sequence of the transmembrane domain of EphA2. Type 7 is highly soluble and interacts with the surface of lipid membranes at neutral pH, while acidity triggers proteination of the glutamic acid residues, which causes transmembrane insertion. Type 7 works as a conditional transmembrane domain where membrane insertion is triggered by a pH drop. Type 7 strongly binds to endogenous EphA2 in cells. Interestingly, the presence of EphA2 facilitates the membrane insertion of type 7. Once type 7 binds to EphA2, there is a signaling cascade involving dephosphorylation of AKT that inhibits cell migration as effectively as ephrin A1. However, the molecular mechanism used by type 7 to activate EphA2 involves a different pattern of phosphorylation of the intracellular domains, with large differences in juxtamembrane tyrosine phosphorylation. A second important difference is that while ephrin A1 induces EphA2 to assemble into large clusters, EphA2 bound to type 7 forms instead medium-sized oligomers. Taken together, our results show that type 7 inserts into membranes where it interacts with the transmembrane and juxtamembrane region of EphA2. These interactions activate EphA2, and the activation loop of the kinase domain is phosphorylated, but not the juxtamembrane region. Type 7 binding also causes oligomerization of EphA2. These results can have important implications for the design of new pH-triggered ligands that modulate protein-protein interactions in other membrane proteins.